Good evening. This is Dr. Smith. We're going to talk some pre-AP biology tonight, and we're going to be talking about mutations. Gene mutations are mutations that occur on the DNA within a gene that is going to be eventually translated into a protein. Gene mutations are one type of mutation. The other type is chromosomal mutations, which we'll talk about at a different time. The first type of gene mutation that we're going to talk about is a substitution. This is where one base in the codon in the messenger RNA sequence has been substituted with another base, and the consequence. There's three different things that can happen. One is what's called a silent mutation. And what the substitution is involved is here's your messenger RNA. It's the codons are grouped in threes there. Starts with the methionine, ends with the stop. And the second codon here, this is CUA here, it was originally a CUU. This was a uracil. The third base in that codon was a uracil, but it has been replaced by an adenine. Now, in this case, the leucine, or the CUU, codes for leucine. CUU, here we go, codes for leucine, and it's been when the last uracil is replaced by an adenine, CUA. We'll go over here, and CUA also codes for leucine. So even though there's been a substitution, the adenine for the uracil, the amino acid that it codes for, it remains the same. And so the protein never knows the difference. So it's said to be silent. So no harm, no foul. The second type of substitution is one that leads to a missense error. A missense is where the substitution leads to the replacement of an amino acid in the sequence. Okay, and when you change an amino acid in the sequence, you have the potential to change the protein from something functional to something non-functional. So in the second codon again, the CUU, which codes for leucine, the first base has been substituted with the cytosine has been replaced with a uracil. If you go to the codon chart for the UUU, that codes for phenylalanine. So phenylalanine is a very different amino acid than is leucine. So this amino acid sequence might have very different properties, may be non-functional when compared to the original intended amino acid sequence here, this protein. And so this is a missense. It's a missense the term means that you no longer have the same amino acid sequence. The third type of substitution error is what's called a nonsense. That's where the substitution of a base leads to the premature termination of the amino acid sequence. So in the original sequence here, the third codon here, the UGU, codes for cysteine. And the uracil has been replaced by an adenine in this RNA sequence, these codons. UGA is a codon for stop. So if you go over here, UGA is a stop codon. So the consequence of this substitution is that this amino acid sequence stops after the second amino acid. It has the methionine that starts it. The leucine's the second one. But rather than having cysteine, threonine, and then the stop codon, it goes straight to a stop. So this is going to be a shortened, perhaps a very shortened amino acid sequence, most likely, in all certainty, going to lead to a non-functional protein. So a nonsense error is a bad one. So before we talk about the insertion and deletion chromosomal errors, I want to show what's meant by a frame shift. And if we have a series of words here that are all three-letter words, the fat cat ate the rat. Now we can all picture in our minds what the fat cat eating a rat is about. But if a extra F is stuck in here between the F and the A in the word fat, and the codons are still read in groups of three, then the sentence becomes the FFA, TCA, TAT. It's nonsense. These words have no meaning. It's completely different than the original sentence. The insertion frame shift error is very similar to that, only with amino acids. So if we continue using our original messenger RNA here, and we happen to stick in an extra cytosine here between the U and the G in the third codon here, then it becomes UGC, because remember, we're reading it in codons in groups of three. So that C is stuck here at the end where the U used to be. The U now goes over to the beginning of the next codon. So the ACU becomes a UAC. ACC becomes a UAC. So the ACC is still there. It's just being read on different codons now. The consequence of this is that from every amino acid from this point on is going to be different than what it originally started out to be. So instead of being a cysteine, threonine stop, it becomes a cysteine, tyrosine, leucine, and who knows what as the messenger RNA, the codons continue on here. So the consequence of this is that from this point forward, from the cysteine amino acid on, the amino acids are completely different. These yield non-functional proteins, essentially non-usable proteins, and these are often associated with lethal consequences. The frame shifts are bad because every single amino acid after the in this case the insertion is going to be the wrong amino acid, and without the proper amino acids, you don't have the proper form, you don't have the proper function. That's an insertion. Now, if you delete a letter, and we go back to our sentence here, the fat cat ate the rat. If you delete a letter, the same thing happens. Instead of the fat cat, we have the ATC, ATA, so forth, so on. It's missing that F there. This means nothing. And very similarly, the proteins, the amino acid sequences will mean nothing. So if we delete a base, and in this case, deleted this U here, the next codon becomes the GUA. Then, since it's read groups of three, it's CUA, CCU, GA, and so forth and so on. We can't see those, we don't know, but the point being that instead of having a cysteine, threonine stop, you have a valine, leucine, proline, and it may continue on for thousands of amino acids before it happens to run into a now stop codon. This is a deletion frame shift error, and like insertion frame shifts, these are very bad. Never have a good outcome because the amino acid sequence is completely changed from the point of the deletion on, and that protein will be non-functional, potentially lethal. All right. I hope that was helpful.